In class 11b, we'll discuss the mechanisms of protein synthesis, and we'll have initiation of prokaryotic and eukaryotic protein synthesis, elongation and termination, antibiotic inhibitors of protein synthesis, and coupling of transcription and translation. So first we'll talk about the mechanism of peptide chain elongation. And on our ribosome, we're going to have an A site, and this is going to be the incoming amino acyl tRNA. And then we're going to have in the P site the peptidal tRNA, and the electron pair of the amino group of the amino acid that's attached to the tRNA will react with the carbonyl group that's attached to the 3' OH. And when we do that, we transfer this entire group over to the tRNA that's now in the A site. So you notice we've added this particular group onto the end of the amino acid, and now the tRNA that was in the P site no longer has a peptide or an amino acid on it, and it's going to dissociate. So we go from a peptidal tRNA and amino acyl RNA to an uncharged tRNA and a peptidal tRNA. So that's basically the mechanism, and then we're going to move the ribosome. So elongation follows a three-step process. So let's start where here's our ribosome, and here's 3 sites. We have an A site, a P site, and an E site. And at this point, the P site is occupied by a transfer RNA that has a growing peptide chain. We have codon recognition, and an incoming amino acyl tRNA recognizes the triplet codon. The binding of this tRNA, amino acyl tRNA, requires a high energy bond from GTP, and we hydrolyze to GDP. Here it says 2 GTPs. It's actually 1, so it's 1 GTP rather than 2. And now we have a tRNA with a peptide chain on it in the P site, and we have an amino acyl tRNA that's in the A site. And again, the amino group here reacts with the carbonyl group here. And now we've transferred this entire peptide chain to the tRNA that's in the A site. This is peptide bond formation. And now we have translocation, and that requires GTP. The amino acyl tRNA that has the peptide on it is now found in the P site. The A site is empty, and the uncharged tRNA occupied the E site, and now it dissociates. And we're ready for the next amino acyl tRNA, which will come in and recognize its codon, and then we will do the elongation, etc. So this will go on until we encounter a termination signal. A more detailed 3-site elongation mechanism is scanning electron microscope data of what the ribosome looks like. So that's what these are. The blue color represents the large ribosomal subunit. The yellow color represents the small ribosomal unit. The green represents the tRNA that has the peptide chain on it. This color represents the tRNA that no longer has the peptide chain. E and E, P and P refer to, the top letter refers to what's happening in the 50S ribosome. The bottom letter refers to what's happening in the 30S ribosome. So this site, the A site, is empty. So an amino tRNA plus elongation factor tau that's bound to GTP will bind to the A site, and it only binds to the 30S part of the A site, because the 50S is blocked because this contains tau. So here's the tau part, EF tRNA, and notice it's not binding entirely to the A site, so that's why they have it off to the side like that. And when this binds to the A site, the uncharged tRNA dissociates. So now we have an E site that's not binding a tRNA. Elongation factor tau, it hydrolyzes GTP to GDP plus inorganic phosphate, and it dissociates. And now both this tRNA and this tRNA with the peptide, this represents an amino acid, these balls represent the peptide, bind entirely to the P site and the A site in both the 50S and the 30S. We have transpeptidation. That's the peptidal transferase activity of the 50S subunit, transfers this peptide group over to here. So remember, the amino group of this amino acid reacts with the carbonyl over here, and now we have the growing peptide chain, and it's on the tRNA in the A site. The P site now has an uncharged tRNA, and now we start to move the ribosomes. So what this means, where you see it slanted like this, and part of the tRNA is in an A site, part is in a P site, this one's partly in a P site, partly in the E site, and now we have an elongation factor G that binds GTP. And when we do that, we get our final translocation, so the ribosomal subunits move. The 30S moves independently of the 50S. So now the 50S moves. We're now looking at the next codon. So the A site is now empty. The ribosome has moved one codon down the mRNA. It would be nice if this actually showed the mRNA right here, but it doesn't. And now the A site is empty. The E site is uncharged. The P site has the aminopeptidyl tRNA, and now a new RNA with elongation tau will come in, and it'll bind to this site. And which one binds? The one that recognizes the codon. And then we go through this process over and over again, and there are proteins that have over 1,000 amino acids, so we would go through this particular phenomenon 1,000 times to get a protein of 100,000 molecular weight. So this is a nice picture in more detail than our other picture, but it shows essentially the same thing. Termination of translation. So here's our 30S ribosomal. Here is our 50S ribosomal. Here's our last tRNA. So this structure is more like the three-dimensional structure of a tRNA. Notice how it looks like an L. So when we encounter a stop signal, remember there's three of them. This is showing UAA. And we have a ribosomal release factor and an elongation factor G that has GTP on it. And when the release factor binds to the UAA, it will hydrolyze off the polypeptide. It will release the tRNA. The release factor is released. GTP is hydrolyzed to GDP. Here's the mRNA that's released. And in this particular picture, it shows that we maintain our inactive 70S ribosome. That's actually controversial. Most references say that these two will dissociate so that we can reassociate them on a new messenger RNA. So keep that in mind. Depending on your reference, it'll show it either coming off as a 70S or it will show dissociation into a 50S and a 30S. Here's another version of termination. Here's our polypeptide chain that's growing on the tRNA. Here's our stop codon, and we have several. We have a UAG, UAA, UGA. Then we have a release factor. The release factor binds, and it hydrolyzes off this peptide. So the peptide gets released. The tRNA gets released. So here's the free peptide. Here's a tRNA being released. Here's a release factor. This doesn't show the elongation factor with the GTP. So release requires hydrolysis of one GTP. And now, upon release of the peptide, the release of the tRNA, all of these things dissociate into the release factor, the 30S subunit, the 50S subunit, and the messenger RNA. So this shows a little bit different picture than what the picture showed before. When one of the three possible stop codons is reached, a release factor is bound instead of a tRNA. Release factors add water to the bond, holding the last amino acid to the last tRNA, hydrolyzing the bond, and releasing the polypeptide. The last tRNA is released, and the translation assembly comes apart. Antibiotics that disrupt protein synthesis. So this gives you a list of antibiotics. Some of these are common antibiotics that you've heard of before, and this is their action. And the action is going to be how it affects protein synthesis and on what ribosomal subunit does it affect protein synthesis. So streptomycins and other aminoglycosides, so these are called aminoglycoside antibiotics. They inhibit initiation and cause misreading of mRNA, and this happens in prokaryotes. Tetracycline, which I'm sure you've all heard of, binds to the 30S subunit and inhibits binding of amino acyl tRNAs, and that's in prokaryotes. Chloramphenicol inhibits the peptidyltransferase activity of the 50S ribosomal subunit, and that's in prokaryotes. Cyclohexamide inhibits the peptidyltransferase activity of the 60S ribosomal subunit, and that's in eukaryotes. So we can inhibit peptidyltransferase with chloramphenicol. That will kill bacteria. We inhibit the peptidyl-transferase activity of the 60S with cyclohexamide. That will kill eukaryotic organisms, yeast, fungi, etc. Erythromycin binds to the 50S subunit and inhibits translocation. So when this binds, the 50S subunit doesn't translocate, and puromycin causes premature chain termination by acting as an analog of amino acyl tRNAs, and it works in prokaryotes and eukaryotes. So here's tetracycline. Here's our 4 rings. So tetracycline. Chloramphenicol, remember the chloramphenicol, works at peptidyltransferase of prokaryotes. Puromycin's claim to fame is it looks like the acceptor arm of a transfer RNA. So here's a transfer RNA. So here's the transfer RNA part. Here's a phosphodiester bond to the adenine that's part of the CCA on the 3' N. Here's the ester bond between the 3' OH group and the amino acid tyrosine. So this is the tRNA that carries tyrosine. And then over here somewhere is the anticodon. And here's puromycin. This part looks like tyrosine. Notice that looks like tyrosine, except it has a methyl group on the O. This part right here looks like the amino acid, but rather than being bound to an O, it makes an amide bond rather than an ester bond. And then this looks like adenine, but it's dimethylated, so the nitrogen has 2 methyl groups on it. And the OH part right here is not attached to a tRNA, so this looks like the very 3' N of a tRNA with the adenine and the amino acid attached, but it doesn't have the rest of the tRNA molecule. So how would this work? So disruption of peptide bond formation by puromycin. So here's the P site, and this has our tRNA. This is the growing polypeptide chain right here. Here's the tRNA. It will be binding to a codon, so we can see it. Right here's the tRNA. It's interacting with the codon in the P site. Here's the E site. And now puromycin binds, and notice it doesn't have the rest of the tRNA bound to it, so it binds to the 50S ribosomal subunit in the binding site for this part of the molecule, but it doesn't bind in the 30S where we have the triplet codon. So the amino group of puromycin will react with this carbonyl group, and it'll make a growing polypeptide chain, but there's nothing to keep the puromycin polypeptide chain attached to the A site, and this dissociates, and that prematurely stops polypeptide chain formation. So puromycin acts like a tRNA on one part of the molecule, but on the other part of the molecule, it doesn't attach itself to the mRNA and bind to the A site on the 30S ribosomal subunit. And as soon as we get our reaction, the polypeptide chain dissociates, and typically this polypeptide chain will be worthless vis-a-vis its regular biological function. Coupling of transcription and translation in bacteria. So here's our RNA polymerase. Here's our double-stranded DNA. Transcription bubble. This would be the 5' N, which means this is the 3' N. So this is the 5', this is the 3'. This is the chain that's growing. This is the mRNA that's being synthesized. And when the mRNA gets long enough, ribosomes can bind to it and start to synthesize the polypeptide chain. So this is the direction of translation. This is the direction of transcription. So as we make the mRNA, we can start to translate it into protein. And notice this ribosome has traveled farther down the chain than this ribosome. And the polypeptide is longer in this one than in that one. It's read more codons. It has a longer polypeptide chain. So this is called coupling. So we can transcribe and translate at the same time. And remember, this is prokaryotes. Eukaryotic transcription and translation happen in different compartments and cannot happen simultaneously. Here we're simultaneously synthesizing protein as we're synthesizing the messenger RNA. But here we make the messenger RNA and it has to be processed. We have to do the splicing, we have to add the cap, we have to add the poly A tail, and then we translocate it through the nuclear pores, and it's at that point that it associates with the ribosome. Here's our ribosomal subunits, and they associate with the mRNA. And now here we're getting protein synthesis. Protein synthesis doesn't happen in the nucleus in eukaryotes. It happens in the cytoplasm, and that means that these aren't coupled, they're uncoupled. So the synthesis of the mRNA is uncoupled from protein synthesis. So it's only in prokaryotes that we can transcribe and translate the same mRNA at the same time. So here we see our amino acyl tRNA synthetases. Those are in the cytoplasm. Here is the ribosomes. They're in the cytoplasm. The cap, the introns, the poly-A, that all happened in the nucleus. So we've uncoupled mRNA synthesis from protein synthesis. And now formation of polysomes. The mRNA can bind multiple ribosomes. We need about 30 nucleotides between each ribosome, and we can have 10 or 15 or 20 ribosomes on one mRNA, and they're all synthesizing the polypeptide chain. And notice the farther the ribosome is along, the more chain we have. When we get to the termination signal, these dissociate. Here, this is our folded protein. This one's about to hit the termination signal. It will dissociate, release the protein. So it's like a factory. This is a production line. And this is what a polysome looks like under the electron microscope. So these are called polysomes. And this can happen in both eukaryotes and prokaryotes. So this particular picture is represented by this cartoon. And here's the ribosomes. And you can see from the ribosomes, you can actually see the proteins being synthesized. So this is part of a protein, that's part of a protein, and that's different from the mRNAs. You can see the mRNAs running down the middle, but the globules coming off, that's the protein as it's being formed, the growing polypeptide chain. So this completes Lecture 11b. Our next and final class will be on protein sorting, which is an extension of what we're doing right here. So protein sorting could just as well be called L11c rather than L12, but just for simplicity, we're going to call it Lecture 12.